So today I'm going to talk to you about Bordetella pertussis evolution in the UK over the last century. So Bordetella pertussis is a bacteria that causes whooping cough exclusively in humans, and we've had a vaccine for this since the 1950s. This was replaced in 2001 with an acellular vaccine, a different type of vaccine that includes 4 to 5 purified antigens from the bug. But since this change, we've seen waning immunity to this vaccine over time and multiple outbreaks like the one we are currently experiencing at the moment. So we wanted to look at how this change, how this bug might be adapting to the vaccines that we use. And to do that, we need to look at the genomes. But up until the advent of long-read sequencing, this was quite hard in pertussis because it has over 240 copies of an insertion sequence, which is over 1 kb long. So short-read sequences weren't able to resolve these genomes. And so we missed the extensive genomic variation that pertussis has, because it doesn't really do SNPs, and it doesn't go in for horizontal gene transfer either. So its main mechanism of variation between species is deletion of genes between these regions. And so this was part of a larger project of work where we were sequencing older strains to capture the full trajectory of pertussis evolution over time and how it might be adapting to the vaccine. And I took this data to conduct a pangenome analysis to look at what genes are present in what genomes and has there been a change. And so, as I said, long-read sequencing and nanopore technology is absolutely vital for this species. And so I used the latest V14 chemistry and flow cells to sequence all our strains that we were sent from the UK Health Security Agency. I was able to assemble high-quality, single-config, nanopore-only assemblies and pass these to our pangenome analysis tool, ggCaller. And this was able to output a presence-absence matrix and a bunch of other stuff to look at what genes are in what genomes. So this is just a snippet from the matrix, but we've got genes down the side and the genomes that they're in across the top. And as expected for pertussis, it's highly conserved a lot of the genes with very few regions of difference. But one in particular stood out, this region of difference, which I term ROD-D. And when we look at the pangenome neighborhood joining tree, which is clustering strains based on the genes they share in their pangenome, we see that region of difference D is largely responsible for this clustering. And we also see that it is exclusively, almost, isolated in strains from the pre-vaccine and whole-cell vaccine eras. But also, interestingly, in the lab, these strains seem to grow faster. And so we wondered, is that significant? Should we look into that? And yes, if you're a pertussis and you have this region of difference, which is about 24 kb, you're more likely to grow faster and be isolated in the older era. And so in conclusion, we were able to sequence high-quality nanopore-only assemblies for pertussis, find this region of difference which might be important for growth, and these were isolated in the pre-vaccine, whole-cell vaccine era, begging the question whether this has been lost and we are selecting for slower growth. And what that might do, we don't know. So the next steps, we're knocking that region out of the old strains, knocking it into the new strains, and assessing if it has the effect on growth that we think it does. And so, thank you very much.